Hey guys, so a classmate asked, what are transposons and why are they important? The formal definition of transposons is that they are DNA sequences that move from one location on the genome to another. The verb transpose can be derived from this name, which means to change the relative place or normal order of something, with transposons also being called transposable elements, TE, or jumping genes. Barbara McClintock first identified these elements more than 50 years ago, although to the initial skepticism of her peers. For much of the 20th century, genes were considered to be stable entities arranged in an orderly linear pattern on chromosomes, much like beads on a string. McClintock's description of mutations that switch genes on and off was at odds with the existing idea that mutations permanently inactivated genes. Her studies of chromosome breakage in maize led her to discover a chromosome breaking locus that could change its position within a chromosome. She identified a particular chromosome breakage event that always occurred at the same locus on maize chromosome 9, which she named the DS, or dissociation, locus. McClintock discovered that DS could change position within the chromosome, as additional experimentation revealed that chromosome breakage at this locus required a second dominant locus. McClintock named this locus activator, or AC, and found that DS chromosome breakage could be activated by an AC element at a different site, or even on a different chromosome. She summarized her data on the first discovered transposable elements in a 1950 classic article called The Origin and Behavior of Mutable Loci in Maize. It was not until the 1980s that AC and DS transposons were molecularly cloned and isolated, with confirmation that transposons were widespread among eukaryotes, eventually leading to the wider appreciation of McClintock's original discovery. Finally, in 1983, McClintock was awarded an unshared Nobel Prize in Physiology or Medicine for her discovery of mobile genetic elements. Today, scientists know that there are many different types of TEs, as well as a number of ways to categorize them. One of the more common divisions is between those TEs that require reverse transcription, the transcription of RNA into DNA, in order to transpose, and those that do not. The former elements are known as retrotransposons, or class 1 TEs, which rely on the enzyme reverse transcriptase to make a DNA copy of the RNA to insert in a new location. Class 2 TEs, or DNA transposons, rely on a different enzyme, transposase, for the DNA to move directly from place to place. The ACDS system that McClintock discovered are classified as Class 2 TEs. Class 2 transposons move by a cut-and-paste process, where the transposon is cut out of its location and pasted into a new location. Transposase binds to both ends of the transposon, which consist of inverted repeats, which are identical sequences reading in opposite directions. They also bind to a sequence of DNA that makes up the target site. Some transposases require a specific sequence as their target site, while others can insert the transposon anywhere in the genome. After the transposon is ligated, or joined by molecular fragments with a new chemical bond to the host DNA, the gaps are filled in by Watson-Crick base pairing. This creates identical direct repeats at each end of the transposon. Note that DNA transposons never use RNA intermediaries. They always move on their own, inserting or excising themselves from the genome by means of this cut-and-paste mechanism. Unlike Class 2 TEs, however, Class 1 TEs do move to the action of RNA intermediaries, with the Class 1 TEs producing RNA transcripts and relying upon reverse transcriptase enzymes to reverse transcribe the RNA sequences back into DNA, which is then inserted into the target site. Both Class 1 and Class 2 TEs can be either autonomous or non-autonomous. Autonomous TEs can move on their own, while non-autonomous elements require the presence of other TEs in order to move. This is because non-autonomous elements lack the gene for the transposase or reverse transcriptase that is needed for their own transposition, so they must borrow these proteins from other elements in order to move. The AC element, for example, are autonomous because they can move on their own, whereas DS elements are non-autonomous because they require the presence of AC in order to transpose. Much of what a transposon does depends on where it lands. Landing inside a gene can result in a mutation, as was discovered when insertions of line 1 or L1, a class TE, were found in APC, a tumor suppressor gene for colon cancer cells, but not in the APC genes in healthy cells in the same individuals. But not all transposons jumping results in deleterious effects. In fact, transposons can drive the evolution of genomes by facilitating the translocation of genomic sequences, the shuffling of exons, and the repair of double-stranded breaks. However, most TEs appear to be silent, not producing a phenotypic effect or actively moving around the genome. Some silenced TEs are inactive because they have mutations that affect their ability to move from one location to another. Others are perfectly intact and capable of moving, but are kept inactive by epigenetic defense mechanisms, such as DNA methylation, chromatin remodeling, and miRNAs. Therefore, the ability of transposons to increase genetic diversity, together with the ability of the genome to inhibit most TE activity, results in a balance that makes transposable elements an important part of evolution and gene regulation in all the organisms that carry these sequences. I hope this video helped you to learn more about transposable elements and their importance.